Okay, let's unpack this. We are diving into really the molecular architecture of your reality. Welcome back to the deep dive.
It's good to be here.
Today, we're sort of putting aside the grand physiology of perception and going straight to the core biochemistry. The fundamental question: how does an external signal, a sound waves, a photon, a tiny molecule, get converted into, the universal language of your nervous system?
It really is the fundamental question of biological life, isn't it? We're exploring how organisms achieve these astounding feats of sensory detection.
Like what? What are we talking about?
Well, like distinguishing between thousands of different chemicals with your nose or your eye registering just a handful of photons. The sensitivity and the specificity required are just breathtaking, and it all comes down to chemistry.
And I find the starting premise so powerful. The idea that our body didn't invent five totally separate systems from scratch. It repurposed components we already use for internal cell communication.
Molecular economy at its finest. Precisely. If you look across the five major senses we'll be talking about, olfaction, taste, vision, hearing, and touch, you just see the same two main players adapted brilliantly over and over again.
And those two players are the core of our toolkit for this deep dive.
They are. The first, and you could argue the most versatile, are the seven-transmembrane receptors, the 7-TMs. These are your long-distance communicators, typically coupled to what we call G proteins. They specialize in really complex chemical detection or light detection. They excel where amplification and specificity are absolutely critical, even if they're a little bit slower.
Okay, so that's player one, the complex, slower system, and the second player.
The second are the ion channels. Much simpler. These are just gates in the cell membrane. They specialize in speed and mechanical detection. They're all about instantly converting physical force or the presence of simple ions into an electrical signal.
So together, these two mechanisms translate every single environmental signal into either a depolarization or a hyperpolarization, which is what forms a nerve impulse.
That's the whole game.
That's our mission today, then, to systematically explore the specialized biochemical mechanisms behind each of the five major mammalian senses. We're going to see how these slow, nuanced 7-TM pathways contrast with the instantaneous speed of the ion channels. So let's start with the one that gives us the most complex chemical picture of the world: olfaction.
The sense of smell.
Olfaction. It provides such nuanced information, doesn't it? It tells us if food is ripe, if danger is near, or it can even link us so powerfully to specific memories. You mentioned earlier that we can distinguish thousands of distinct chemical compounds. What exactly defines an odorant from a biochemical standpoint?
An odorant is almost invariably a small, volatile, organic compound. It has to be. It needs to be small enough to be carried as a vapor, then it has to cross the mucus layer in your nasal cavity, and finally bind to a receptor.
Okay, so small and floaty.
Exactly. And the sources give us some great examples of this chemical specificity. Think of benzaldehyde, which is the primary molecule giving you that distinct almond odor. Or, on the other end of the spectrum, 3-methylbutane-1-thiol. That's the infamous molecule responsible for the overwhelming odor produced by skunks.
Right, so very specific molecules. But is there a common chemical feature they share? I mean, are they all hydrophobic, for instance? Is there a general rule?
That was an early thought, that there might be some simple physical property. But the crucial finding that really overturned those theories is that the shape of the molecule is the critical determinant of its smell. It far outweighs general properties like size or hydrophobicity.
The three-dimensional shape. How do scientists actually prove that shape is the key factor?
The classic evidence comes from enantiomers.
Mirror image molecule.
Exactly. They're molecules that are perfect mirror images of one another, like your left and right hand. They have identical chemical formulas. They share almost all physical properties, boiling point, solubility, all of that.
But they smell different.
They can smell profoundly different. The source material notes the fascinating difference between the molecules for spearmint and caraway. One smells sharp, minty. The other smells, herbaceous and savory, like in rye bread. They are perfect mirror images, but the different orientation of their atoms means they must interact with the receptor surface in a completely distinct way.
That immediately suggests it's not just a general interaction, it's a tight, three-dimensional fit. It sounds like a highly specific protein receptor binding site, a lock and key.
It absolutely necessitates that conclusion. The odorant is interacting with a specific protein receptor, And we see even more evidence for this specificity in clinical observations, particularly something called specific anosmias.
Specific anosmias.
Hmm.
So the inability to smell just certain compounds, that's a compelling piece of evidence. How does that link back to the receptor structure?
Well, these are often inherited conditions. An individual can be perfectly capable of smelling everything else, roses, coffee, smoke, but they cannot smell one or two very particular compounds, like certain steroids or musk compounds.
And if your whole olfactory system is working fine, but you're missing one specific smell.
It strongly indicates a mutation in the single gene that encodes the specific receptor protein meant to recognize that unique molecule. It's definitive proof that there's a dedicated molecular sensor for many of these odorants.
So if these receptors are the key, where are they? Where are they located anatomically, and how vast is this receptor system in mammals?
The detection all happens in the main olfactory epithelium. It's a small, very specialized patch of tissue right at the top of your nasal cavity.
Okay.
And this epithelium houses about 1 million sensory neurons. The actual receptor proteins, the sites where the odorant binds, are on these fine, hair-like projections called cilia that stick out from the neurons into the mucus layer.
1 million neurons.
Yeah.
And genetically, what family do these receptors belong to? They have to be incredibly versatile.
They belong, unequivocally, to the 7-TM-helix receptor family. That same family we mentioned at the start. It's an enormous family, but the subset responsible for smell, the odorant receptors, or ORRs, is exceptional. The human genome contains approximately 350 functional ORR genes.
350. So that means 350 different flavors of the same basic 7-TM receptor structure.
That's right, and that's a significant number of functional genes. But the source mentions something even more interesting about our evolutionary past.
The pseudogenes.
The pseudogenes, yes. We also have about 500 OR pseudogenes in our genome. These are just genomic relics. They're non-functional genes that have picked up mutations over time that prevent them from producing a properly folded or functional receptor protein.
So why does that ratio 350 functional to 500 non-functional? Why does that matter?
Well, it tells a fascinating story about our evolution. When researchers look at the genetics across different mammals, they find that the fraction of these pseudogenes dramatically increases in species that are more closely related to us, to humans.
So compare us to, say, a mouse.
A mouse or a dog or a rat. They're highly dependent on olfaction for, navigation, finding foods, social cues, and their genomes have far fewer pseudogenes. Nearly all of their OR genes are functional and ready to go.
So as higher mammals like primates develop better vision and maybe larger brains for processing other information, the selective pressure on smell acuity decreased.
It must have. And many of those genes they fell into disuse. We basically sacrificed some of our sniffing power for other traits. It's a powerful connection between genetics and behavior written right there in our DNA.
That's incredible. So structurally, how do these 350 receptors manage to bind thousands of unique shapes? They all share that basic 7-helix architecture.
They do. They share conserved regions that are necessary for the internal cell signaling part, but they must have hypervariable regions for the binding part.
Right.
And that's exactly what we see. Like other 7-TMs, they have strongly conserved amino acid sequences, particularly in the loops that interact with the G protein inside the cell. But critically, they have these highly variable regions.
And that's the binding pocket.
That's where the binding pocket is. The sources strongly suggest it lies within that hydrophobic pocket formed by the 7 helices, and they specifically highlight transmembrane helices 4 and 5 as the sites of maximum variability.
Which makes beautiful biochemical sense.
Mm-hmm.
The highly variable regions are what define the shape of the lock, allowing for hundreds of variations on the same receptor theme.
Precisely. So now let's track the signal. Once an odorant binds to that specific variable site, how is that chemical recognition converted into a neuronal impulse?
We know it's a G protein cascade. What was the first key piece of evidence that pointed in that direction?
The definitive clue came from early biochemical studies. They found that exposing the olfactory epithelium to odorants caused a measurable increase in the intracellular levels of cyclic AMP.
Okay, a second messenger.
But crucially, this only happened if GTP was also present in the mixture, and that requirement for GTP is the molecular fingerprint of a G protein interaction. The receptor has to activate a G protein to get it to exchange its GDP for a GTP.
Perfect. So what's the specific G protein involved here, and what's the exact cascade?
The specific G protein is unique to the olfactory system. It's designated G-olf for olfactory.
Created.
Very. And the cascade involves 4 precise steps. First, the odorant binds to its OR, causing the 7-TM receptor to change conformation.
The activation step.
Right. Second, that activated receptor finds a G-ULF protein and gets it to swap its GDP for a GTP. G-ULF alpha subunit then breaks away.
And that alpha subunit is now the active signal. What does it do?
It finds and stimulates a specific adenylate cyclase. That's the enzyme that synthesizes CAMP-P, so you get a huge rapid increase in the intracellular concentration of CAMP-P.
And the final step. How does CAMP cause a nerve impulse?
That sharp rise in CAMP acts directly as a ligand. It binds to and opens a non-specific Hatchery channel.
So it's a CAMP-gated channel. And non-specific eccation means positive ions can rush in.
Exactly. Positive ions, calcium, and sodium flood into the cell. That influx of positive charge depolarizes the neuronal membrane, rapidly generating an action potential. That's the nerve impulse. And it gets sent directly to the olfactory bulb for processing. It's fast, highly specific, and amplified at every single step.
Okay, let's go back to the math problem, though. We have 350 sensors, but we perceive, tens or hundreds of thousands of different smells. This brings us to the combinatorial mechanism of decoding. This is where the true ingenuity lies, isn't it?
It is, by far, the most impressive part of the entire system, and it starts with a foundational principle of expression. Each olfactory neuron expresses only a single OR gene.
Just one, out of 350 possibilities.
Just one. The selection process seems to be largely random, but it's very strictly enforced by a feedback mechanism that suppresses the expression of all other OR genes in that cell. So you have 350 types of sentinels, and each cell chooses to be only one of them.
So if I have 1 million neurons, I have this massive population of highly specialized cells. But no single cell knows the full story of a complex smell.
Precisely. And the genius of it is that there is no simple one-to-one match between a single odorant and a single receptor. The combinatorial logic works in two directions.
Okay, break that down.
First, each unique odorant activates a number of different receptor types, and often to varying degrees. It might activate receptor A strongly, receptor D moderately, and receptor F weakly.
In the second direction.
Conversely, each individual receptor can respond to more than one odorant, as long as the molecules share some degree of structural similarity.
So the brain isn't reading receptor A equals rose. It's reading the specific pattern of activation. Receptor A is on, D is sort of on, F is on full blast, and Z is off.
That resulting unique combination of activated receptors is the chemical code. It's how a limited set of 350 receptors can distinguish an almost unlimited variety of odorants. It's like combining the 26 letters of the alphabet to form millions of words.
But how does the brain maintain a readable, stable map of this incredibly complex pattern? It must get noisy.
It would, except for some incredible neuroanatomical organization. Scientists found that all the neurons expressing the same specific OR gene, say, all the cells that make receptor A don't just scatter randomly in the brain.
They all go to the same place.
They all converge to the same single specific location. It's a little knot of neural tissue called a glomerulus in the olfactory bulb.
So that creates a spatial map of activity. If receptor A is activated by a rose smell, a specific glomerulus lights up. If receptor B is activated, a different specific glomerulus lights up. The brain just has to read the map of lights.
Exactly. It's a spatial map, and it's highly consistent across individuals. If a specific odorant activates a unique combination of 3 glomeruli in your brain, that same pattern of 3 glomeruli will light up in mine. It's a beautifully organized, stable system for chemical coding.
And it's not just a theory, is it?
Yeah.
The sources mentioned that we've validated this with real-world technology.
We have. The combinatorial encoding mechanism is the fundamental principle behind technology, like the electronic nose, for example, the Cyranos 320.
How does that work?
These devices use an array of non-biological polymer sensors. Each polymer binds basically every odorant, but to a different degree, leading to a unique electrical pattern. A small array of just 32 of these sensors, when you couple it to pattern recognition software, can distinguish very subtle chemical differences.
Like what?
Like fresh versus spoiled fruit, or different isomers of hydrocarbons. The biological system simply harnesses this powerful mathematical principle on a much grander scale.
Wow. That dive into olfaction shows this really high specificity combinatorial chemical coding. So now let's move to the complementary chemical sense.
Yeah.
Taste or gustation. It feels much simpler.
It is simpler, at least in terms of discrimination. And of course, smell hugely augments our experience of taste, which is why food tastes so bland when you have a cold.
Right, you block your nose and the flavor just disappears.
But taste itself is limited to sensing only 5 primary, functionally important categories. bitter, sweet, sour, salty, and umami.
And these 5 categories are all about survival and nutrition, aren't they?
Absolutely. They are essential tools for classifying food. Sweet, salty, and umami, which is the taste of amino acids like glutamate and aspartate signal, potentially beneficial or high-energy food sources.
And bitter and sour are the warning signs.
Generally, yes. Bitter often signals alkaloids and plant toxins, and sour can signal spoilage or excessive acidity. They're our first line of defense against ingesting something harmful.
So where does this detection happen on the tongue?
It happens in structures called tooth buds. Each bud is a complex little organ containing about 150 cells, including the sensory neurons, and these neurons have microvilli that project up to the tongue's surface, and they are just rich in the actual taste receptor proteins.
Okay, let's start with the 3 tastes that use that slower but amplified 7-TM receptor mechanism, similar to what we just saw in olfaction. So it's bitter, sweet, and umami. What was the molecular smoking gun that told scientists that G proteins were involved here too?
It was a very similar story to the discovery of G-olf in olfaction. The key clue was the isolation of a specific G protein alpha subunit that was expressed almost exclusively in taste buds.
And they named it.
Gusducin, for gustatory ducin. Its discovery immediately pointed researchers toward the 7-TM receptor family.
Let's tackle the most evolutionarily important protective taste, bitter. How did scientists find the actual bitter receptors?
They used a brilliant combination of human genetics and molecular biology. So the ability to taste certain bitter compounds, like one called PROP, was genetically mapped to a specific spot on chromosome 5.
So they knew where to look.
They knew where to look. Researchers then mined that region of the genome for candidate genes that looked like 7-TM receptors, and that led them to the discovery of the T2R receptor family.
How many of these bitter receptors do humans have?
The human genome contains about 30 functional T2R sequences, and the evidence linking them to bitter taste is very, very solid. They're expressed in the same taste cells as gustducin. And furthermore, specific experiments showed that when a specific bitter compound, like cyclohexamide, was present, it caused GTP to bind to gustducin, but only in cells that were expressing the corresponding T2R receptor. In this case, it was a mouse receptor called MT2R5.
So that confirmed the entire pathway. T2R receptor binds the bitter molecule, which activates gustducin, which binds GTP, and there's your signal.
That's the one. So that's our mechanism for toxicity avoidance. Now let's look at the nutrient-seeking tastes. Sweet and umami. How are they sensed?
Well, sweet taste must be really complex because the receptor has to respond to such a diverse range of molecules. You have simple sugars like glucose and sucrose, but then you have totally different artificial sweeteners like saccharin and aspartame and even some sweet-tasting proteins.
It's a huge challenge. And this broad yet specific recognition is mediated by a specialized 7-TM structure, a heterodimeric receptor complex.
A heterodimer. So 2 different 7-TM subunits have to work together. That's unusual. Which subunits form the sweet receptor?
The sweet receptor complex consists of 2 proteins, T1R2 and T1R3. They're both members of the T1R family, and they're notable because they have much larger extracellular domains compared to the smaller, more compact T2R bitter receptor.
And you need both of them.
You need both. Knockout studies showed that a mouse must express both T1R2 and T1R3 simultaneously to respond to any sweet compound. If you get rid of either one, the entire response is lost.
So T1R2 and T1R3 together form the binding site for all things sweet. What about umami, the taste of protein and amino acids?
Umami, which is triggered by L-amino acids like glutamate and aspartate, is sensed by a very closely related heterodimeric receptor complex. This one is made of T1R1 and T1R3.
Wait, T1R3 is the shared subunit. That's a fascinating piece of molecular economy. T1R3 partners with T1R2 for sweet, and the same T1R3 partners with T1R1 for umami.
Exactly. T1R3 acts as the flexible partner. It's necessary for recognition in both cases. T1R1 is the specific subunit that's responsible for detecting the amino acids.
And you can prove that.
You can. If you disrupt the T1R1 gene in a mouse, that mouse loses its umami response completely, but it will still respond perfectly normally to sweet compounds. It proves the functional distinction of the 2 different heterodimers.
This shared subunit also raises a really profound contrast with olfaction when it comes to the encoding strategy. We emphasize that olfactory neurons express only one receptor type for high specificity. What happens in taste?
And here is the key difference. In taste, the receptor cells that detect bitterness express many different T2R family members all at once.
So instead of one cell specializing in one bitter molecule, The taste cells are generalists for bitterness.
They are generalists.
What's the implication of that lack of specificity at the cellular level?
The lack of specificity in receptor expression leads to a profound loss of specificity in the downstream transmission. When you ingest many distinct bitter compounds, say quinine from tonic water, caffeine from coffee, cyclohexamide from a lab, they all activate different combinations of T2R receptors on the cell surface.
But those signals all converge inside the same sensory neurons.
Correct. So the brain only receives one single message: bitter. We can't chemically discriminate between different bitter compounds the way we can discriminate between, say, the smell of jasmine and the smell of lavender.
Which makes perfect sense for evolutionary necessity. We just need to know if it's potentially dangerous, not which specific danger it is. That's why our taste discrimination is so modest compared to smell.
It's the ultimate expression of structure-serving function. So now we leave the G protein 7-TM systems entirely and move to the tastes that require instant ion sensing: salty and sour. These use direct ion channel detection.
For salty taste, we're fundamentally talking about detecting sodium.
Yes. Salty taste is detected primarily by the direct passage of positive sodium ions, Na plus S, across the membrane, generating a current. And the key player here is the amiloride-sensitive Na plus channel.
Amiloride is that compound that blocks the taste of salt, right?
It mutes it, yes. It accounts for about 80% of the salty taste response. Amiloride physically binds to and blocks the flow of sodium through this 4-subunit ion channel. If the channel is blocked, the influx of positive charge is blocked, the depolarization fails, and the salty taste is muted. The flow of sodium is the signal.
And what about sour taste?
Sour taste is fundamentally the detection of acidity, which really means a high concentration of hydrogen ions, or H+ protons. And the H+ ions achieve their signaling effect by directly interacting with and affecting multiple ion channels at the same time.
How exactly do H+ ions create that sour signal?
They act in a few complex ways. First, H+ ions can actually flow directly through those same amiloride-sensitive Na+ channels we just talked about for salty taste.
Okay.
But second, and critically, they also bind to and interfere with potassium ion channels. By binding to and blocking certain potassium channels from letting potassium out, they disrupt the cell's ability to maintain its normal resting potential.
So the H+ ions aren't just flowing in and depolarizing the cell, they're also trapping other positive charge inside by blocking the exit for potassium.
Precisely. It's a dual attack.
Yeah.
The combined effect, the influx of positive H+ charge and the disruption of potassium efflux, changes the membrane polarization significantly. That leads to the nerve impulse that your brain interprets as the sour sensation. It makes sense for acidity, as it's a strong, potentially tissue-damaging signal.
That switch from the slow 7-TM systems to these fast ion channel systems is the perfect segue to our next major sense, which requires unmatched speed.
Vision.
Vision. We move from chemical detection to electromagnetic detection. This is fundamentally different. It's not relying on a chemical ligand, but on a photon of light to start the whole reaction.
And vision is based on detecting light in the visible spectrum, so from about 390 to 750 nanometers. We have two specialized cell types in the retina to do this. The cones, of which we have about 3 million, handle bright light and color vision.
And the rods.
And the rods, about 100 million of them. They handle dim light and are extraordinarily sensitive. A single rod cell is capable of detecting a single photon.
A single photon. That's incredible. Let's start with those highly sensitive rods and their specialized visual pigment, rhodopsin. We're back in the 7-TM receptor family, but this one is adapted for light.
Rhodopsin is a masterpiece of molecular design. It consists of the protein component, opsin, that's the 7-TM receptor, part linked to the light-absorbing prosthetic group, 11-cis-retinal.
And retinal is derived from vitamin A.
It is. It's a polyene, which is a long chain of alternating double and single bonds, which makes it the perfect molecular light trap.
The efficiency must be astronomical, considering the sensitivity of a single rod cell.
It is. Rhodopsin absorbs light incredibly efficiently, with a maximum absorption peak centered perfectly around 500 nanometers, which just happens to match the peak output of the sun.
That's no accident.
Not at all. And the 11-cis-retinal chromophore is attached to the opsin protein via a protonated shift-based linkage. It connects to a specific lysine residue, lysine 296, which sits in the 7th-transmembrane-helix.
Why is that shift-based linkage so important?
It's crucial for what we call spectral tuning. Free retinal on its own only absorbs UV light around 370 nanometers. When it forms that protonated shift base, the absorption shifts to 440 nanometers. But then the surrounding opsin protein environment fine-tunes it, shifting that maximum all the way to 500 nanometers.
So the protein itself is tuning the antenna.
Precisely. It achieves this optimal tuning by having the positive charge of the protonated shift base stabilized by a nearby negatively charged residue, glutamate 113. This allows the protein environment to dictate exactly which wavelength of light the molecule is most sensitive to.
So the protein is a sophisticated filter. Now, a single photon hits the 11-cis retinal. What is the immediate primary physical event?
The primary event is the conversion of light energy into mechanical force. The photon causes the 11-cis retinal chromophore to isomerize instantly, we're talking femtoseconds, to its all-trans configuration.
A tiny chemical change, a change in shape. But it must result in massive atomic movement within the protein structure.
It does. That isomerization from cis to trans causes the shift-based nitrogen atom to move an estimated 5 angstroms, that's half a nanometer. That violent, rapid atomic movement is the molecular trigger. It kicks off a cascade of conformational changes in the opsin protein, culminating in the fully activated state, rhodopsin II or R star.
And R star is functionally identical to the activated state of any hormone-bound 7-TM receptor we talked about earlier, right?
Absolutely. R star is the active receptor. It's now ready to bind and activate its specific G protein.
Let's discuss that amplification cascade. Which G protein is specific to vision, and how does it translate that photon into the electrical signal?
R star activates the specific G protein called transducin. The cascade is analogous to olfaction in its structure, but its final signaling output is actually the opposite.
Right. In olfaction, the cascade raised cAMP levels and opened a channel. What does the transducin cascade do?
When activated, transducin's alpha subunit goes on to stimulate a crucial enzyme, cGMP phosphodiesterase, or PDE. It turns it on by binding to and removing an inhibitory subunit from the PDE. Once it's activated, the PDE is a hydrolytic powerhouse. It rapidly starts converting cGMP to GMP.
So instead of increasing the cyclic nucleotide, we're rapidly decreasing it.
Yeah.
What does that drop in cGMP do to the cell membrane?
This is the critical part. In the dark, the rod cell is actually depolarized. High levels of cGMP keep these cGMP-gated ion channels open, allowing a constant flow of positive cation sodium and calcium into the rod cell. This is called the dark current.
So in the dark, the cell is constantly signaling.
It is. When light hits, the PDE destroys the cGMP concentration plummets, and those channels slam shut. Stopping that influx of positive ions causes the cell membrane to become more negative. It hyperpolarizes.
That's the remarkable switch. Vision is signaled by the membrane becoming more negative by shutting down a continuous flow of information rather than opening a gate to fire a signal.
Exactly. And because amplification occurs at every single step, one R star activates hundreds of transducins, and each activated PDE hydrolyzes thousands of cGMPs, a single photon is amplified into a powerful, measurable hyperpolarization.
But since we need to see continuously, the system has to terminate and recover incredibly fast. In milliseconds. This complex regulation relies on a brilliant feedback mechanism involving calcium ions.
It does. It has to be fast.
Let's break down that recovery process. First, how is the active R star receptor rapidly shut down?
R star must be deactivated. A specialized enzyme called rhodopsin kinase rapidly phosphorylates the R star at multiple serine and threonine residues on its tail. This phosphorylation acts as a flag.
A flag for what?
A flag for an inhibitory protein called arrestin. Arrestin binds tightly to the phosphorylated R star, and that physically prevents it from activating any more transducin molecules. That effectively turns off the switch at the source.
And the G protein itself needs to be silenced as well.
Correct. Transducin's alpha subunit has an intrinsic GP-pace activity. It's a built-in timer. It hydrolyzes the bound GTP back to GDP, which returns the G protein and the PDE to their inactive states. That silences the signaling pathway.
Okay, so that's the shutdown. What about the final critical step? Restoring the cGMP levels to reopen the channels and bring the rod back to its dark adapted ready state.
This is where the calcium feedback loop takes center stage. The enzyme that synthesizes cGMP from GTP is called guanylate cyclase, or GC, and crucially, the activity of guanylate cyclase is strongly inhibited by high levels of cytoplasmic calcium.
Okay, so in the dark, the cGMP-gated channels are open, which means calcium is constantly flowing in along with sodium.
Exactly. So in the dark, calcium levels are high and GC is inhibited. When the light hits, the PDE destroys cGMP, The channels close, and that calcium influx immediately stops.
But the cell keeps pumping calcium out.
Right. The cell continues to actively pump calcium out of the cell using a plasma membrane exchanger. So the internal calcium concentration plummets rapidly. The sources mention it can drop from about 500 nanomolar down to just 50 nanomolar.
And that sharp tenfold drop in calcium is the molecular signal for recovery.
It is. It instantly relieves the inhibition on the guanylate cyclase. The GC then ramps up synthesis, rapidly restoring the cGMP concentration, which reopens the cGMP-gated channels and completely resets the cell for the next photon.
So light absorption triggers the hyperpolarization, but the subsequent drop in calcium is what triggers the rapid restoration. It's a tight, self-regulating circuit where the ion flow itself dictates the speed of recovery.
It's molecular elegance in action. Now we should shift briefly to color vision, which relies on the cone cells.
Right. The cones use the same chromophore, 11-cis retinal, and the same fundamental 7-TM cascade, but somehow they perceive color.
They do, and that's because cones use 3 distinct photoreceptor proteins. They're all homologs of rhodopsin, but they have different spectral tuning. One peaks at 426 nanometers, which we perceive as blue, one at 530 nanometers for green, and one at around 560 nanometers for red or yellow-green.
Since the retinal chromophore is identical in all 4 rhodopsin and the 3-cone pigments, the difference in color perception must be entirely due to how the protein, the opsin, interacts with that retinal molecule.
That is the astonishing conclusion. The green and red pigments in particular are over 98% identical in their amino acid sequence.
98%.
And researchers were able to pinpoint the entire difference in their light absorption, a shift of 30 nanometers in wavelength, to just 3 specific amino acid residues. Positions 1 eta, 277, and 285.
Only 3 amino acids out of hundreds. That is precision engineering.
It highlights the extreme sensitivity of the chromophore to its immediate molecular environment. In the red pigment, these 3 residues are serine, tyrosine, and threonine, all of which have hydroxyl or OH groups. The green pigment has other amino acids there, like alanine, which lack those OH groups.
And how do those hydroxyl groups create the red shift?
The hydroxyl groups in the red pigment are perfectly positioned to interact electrostatically with the photoexcited retinal chromophore. They stabilize its energetic state. This interaction effectively lowers the energy required for the excitation, which in turn shifts the absorption maximum toward the lower energy, longer wavelength red region of the spectrum.
That's an incredible level of detail. And because the genes for the green and red pigments are so similar, they are prone to a specific genetic mishap.
They are. The genes for both pigments are located right next to each other on the X chromosome. And their high similarity, that 98% identity, makes them highly susceptible to something called unequal homologous recombination during the formation of sperm and eggs.
And this recombination is the cause of most color blindness.
It is the most common mechanism, yes. Recombination can happen between the genes, leading to the complete deletion of one of them. For instance, the green pigment gene gets lost entirely. This results in the most common form of color blindness, where individuals can't easily distinguish between red and green, and it affects about 5% of males because the genes are on the X chromosome.
And what's the second, more subtle consequence of this?
Recombination can also happen within the genes themselves. This can create a hybrid red-green photoreceptor. This new hybrid protein will have an anomalous absorption spectrum, peaking somewhere between red and green. And if an individual has this hybrid receptor but lacks one of the pure pigments, their color discrimination is impaired because the brain can't easily contrast the signals from the 2 necessary color channels.
We've now thoroughly explored the 7-TM pathway for chemical and electromagnetic detection. Let's shift entirely to the world of mechanics with hearing and touch. The fundamental difference here is the need for extreme speed.
Speed is the absolute governing factor in hearing. We are discussing extremely high time resolution. To process sound frequencies up to 20,000 hertz, the system needs to operate on a timescale of 50 microseconds. But what's even more demanding is our ability to localize sound sources.
Right. To locate a sound, you have to detect that minute time difference between the sound hitting your 2 ears.
That interval time difference can be as short as 0.02 milliseconds for precise localization. A G protein cascade, which takes hundreds of milliseconds to activate and hundreds more to reset, it's hopelessly slow. This high time resolution dictates that hearing must use a direct transduction mechanism that converts mechanical motion instantly into ion current flow.
So we bypass all this second messenger's tech MP, cGMP entirely. Where does this lightning-fast conversion happen in the ear?
It happens in the specialized sensory neurons called hair cells, which are in the cochlea of the inner ear. Each of the 16,000 hair cells per cochlea has a bundle of projections called stereocilia, about 20 to 300 of them per cell, and they're organized in this precise length-graded arrangement, like a staircase.
And this graded bundle is the antenna that captures the motion from the sound wave.
It is, and micromanipulation studies confirm the signaling direction. If you displace the hair bundle toward the tallest part, you get depolarization, the positive current signal. If you displace it in the opposite direction, you get hyperpolarization, the negative current signal.
And the sensitivity is just astronomical.
Yeah.
You mentioned a movement of 3 angstroms is functionally important. That's less than the diameter of a single atom. How is that minuscule physical motion linked to an electrical signal?
This is where the anatomy is key. The individual stereocilia are connected to their taller neighbors by these very thin filaments called tip links. These tip links are the physical couplers.
So the tip link acts like a tension spring, a little molecular rope. How does that rope operate the gate?
This is the mechanical gating model. The idea is that the tip links are directly coupled to mechanically gated ion channels located in the stereocilia membrane. You can literally think of the tip link as being physically connected to the channel's gate.
So what happens when a sound pressure wave displaces the whole bundle?
In the resting state, when it's quiet, roughly 15% of the channels are already open, which provides a sort of resting electrical tone. When the bundle gets deflected toward the tallest stereocilium, the tension on those tip links increases dramatically.
It pulls them taut.
It pulls them taut. And that physical tension literally pulls on the channel protein, forcing more channels to open immediately. The instantaneous flow of ions depolarizes the cell, sending the signal.
And if the sound wave pulls the bundle in the opposite direction?
The tip link tension is relaxed. It goes slack. And that causes the open channels to immediately spring shut, which rapidly hyperpolarizes the cell. The entire process mechanical force to ion flow happens in microseconds. No waiting for enzymes to activate or secondary messengers to diffuse.
Has the molecular identity of this extremely fast, mechanically gated ion channel been conclusively identified?
Well, it's been a major area of research, and the focus has really landed on the TRP transient receptor potential channel family. The search often starts outside of vertebrates. For example, in the fruit fly Drosophila, they have these mechanosensory bristles that detect air currents, and researchers identified a TRP channel protein there called NOMPC.
And what does the structure of NOMPC tell us about how it translates a physical pull into an open pore?
The structure is highly suggestive. One region of NOMPC forms the ion channel pore, as you expect, but the crucial N-terminal region consists almost entirely of 29 anchor-in repeats.
Anchor and repeats, what are those?
They're common structural motifs, a hairpin loop followed by a helix-turn helix, and they're known primarily to mediate very rigid protein-protein or protein membrane interactions.
So the anchor and repeats aren't just anchors, they're the rigid scaffolding. The spring mechanism that physically couples the tension from the tip link to the structural change needed to open the channel pore.
That is the leading hypothesis. They provide the necessary stiffness and coupling mechanism. And in vertebrates, a highly promising candidate is a channel called TRPA1. It's also a TRP channel, and it contains 17 anchor-in repeats. And it's expressed specifically in the tips of vertebrate hair cells, which strongly supports the idea that this structural motif is a universal solution for converting nanometer-scale displacement into electrical current.
The elegance of this TRP channel family is really starting to become clear. They're used for speed and physical sensing, whereas the 7-TM receptors handle the complex chemistry.
A very clear division of labor.
That brings us to our final sense. Touch. And like taste, touch isn't really a single sense, but a combination of systems detecting pressure, temperature, and pain across the skin. We noted briefly that pressure detection is thought to involve ion channels homologous to those in salty taste.
Yes, the mechanosensory neurons that are responsible for sensing pressure appear to utilize those amiloride-sensitive sodium channels, similar in structure to the ones that detect salt, which again just illustrates that molecular repurposing theme. But the most profound biochemical findings in the sense of touch really relate to pain detection.
Pain is transmitted by specialized neurons called nociceptors, which warn us of actual or potential tissue damage. The discovery here relied on a surprising chemical connection.
A connection that most people experience in their kitchens. Capsaicin.
From chili pepper.
From chili peppers. Capsaicin is the molecule that gives them their hot taste. But when you eat it, it doesn't just taste hot. It actually activates the nociceptors in your mouth, causing a true pain response. And this ability to trigger pain chemically provided the perfect molecular bait for researchers.
So researchers used capsaicin to hunt for its receptor. How did they isolate it?
Well, they hypothesized that capsaicin must be opening an ion channel in these nociceptors. So they looked for neurons that showed a rapid uptake of calcium only when they were treated with capsaicin. This molecular fishing expedition led them to the identification and cloning of the receptor, which was named VR1 for vanilloid receptor 1.
And what did VR1 turn out to be?
Another crucial member of the TRP channel family. The active VR1 receptor is a tetramer, 4 units assembled together, and like its cousin TRPA1 in hearing, it also contains 3 ankyrin repeats in its amino terminal region. So that recurring structural motif strongly suggests that VR1 is also capable of interacting dynamically with structural elements inside the cell.
The most astonishing thing about VR1, though, is that it isn't just a capsaicin receptor. It functions as an integrator of noxious stimuli. It's like a single molecular gatekeeper that just screams danger when it's activated by 3 very different forms of harm.
That integrative function is its most important survival mechanism. VR1 is a cation channel that can be activated by, first, capsaicin at submicromolar concentrations. Second, by high temperatures, specifically any temperature above 40 degrees Celsius, so thermal heat pain. And third, by dilute acid. A drop in pH below 6.0, often down to a midpoint of 5.4, is sufficient to open it.
So if you accidentally put your hand on a hot stove, the heat opens the VR1 channel. If your tissue is inflamed due to an injury, the local acidity opens the VR1 channel. And if you handle extremely hot chili peppers, the chemical capsaicin opens the VR1 channel.
Yes. And critically, it integrates these signals. The response to heat, for example, is significantly greater when the local tissue pH is lower, more acidic. VR1 basically pulls these different types of destructive energy, chemical, thermal, and acid stress into a single nerve impulse, which leads to the perception of pain and triggers immediate protective reflex actions.
That's molecular intelligence. Now let's explore the clinical application it's mentioned in the sources. Capsaicin is used in pain management, even though it causes pain initially. How does that paradox work biochemically?
It's a mechanism of therapeutic desensitization. When nociceptors are stimulated continuously by capsaicin, they are initially overwhelmed, which causes severe pain. However, chronic or prolonged exposure to capsaicin overstimulates these pain-transmitting neurons, and over time, this overstimulation leads to a state of desensitization in the neurons, making them far less responsive to subsequent pain stimuli.
So you're using a mild topical burn to chemically exhaust the pain transmission machinery.
Effectively, yes. This dampening effect makes topical capsaicin a really valuable tool in managing chronic pain conditions like arthritis or neuralgia, where the whole goal is just to quiet down persistently overactive pain pathways.
It's a remarkable way to illustrate the principle of homeostasis. If you push a biological system too hard for too long, it shuts itself down to protect itself.
That's a great way to put it.
This deep dive has been a comprehensive tour through the 5 senses, showing us that the difference between seeing light and hearing sound is, at its core, simply a change in molecular mechanism. The world we perceive is entirely structured by the shape of proteins and the flow of ions.
It is, and we've synthesized the 2 main molecular strategies that really define our interaction with the external environment. First, there's the complex 7-TM receptor G protein cascades. These are the slow and specific systems used for vision, olfaction, sweet, bitter, and umami taste. They're all about maximizing amplification and chemical discrimination, often using that clever combinatorial coding.
And second, the fast and direct approach. The direct mechanical gating of ion channels. This is used where speed is absolutely essential in salty and sour taste, in hearing, and in the integration of pain and touch signals. These systems convert pressure, temperature, or ion concentration changes instantly into electrical current.
And the overarching biochemical theme is undeniable: molecular repurposing. Our cells did not need to invent entirely new classes of signaling molecules for perception. They just took the existing machinery, 7-TM receptors, G proteins, cyclic nucleotide regulation, and adapted them brilliantly to process photons, vibrations, and molecules from the environment.
It provides such a complete mechanistic picture of perception. We understand how chemical energy and physical energy are converted into those electrical signals, the depolarization and hyperpolarization that travel to the brain.
But every complex system like this requires energy just to maintain a state of readiness. We focused on the signal flow, but consider the hidden work. For a rod cell to be poised to detect that single photon, or for the auditory hair cell to be ready for the next microsecond of sound, they have to constantly maintain these steep ion gradients across their membranes, often working against equilibrium.
That's a fascinating layer of complexity we often overlook.
Or think about vision, the chemical energy that's required to rapidly recycle the all-trans retinal back to the essential 11-cis retinal so that you can see continuously.
So what biochemical mechanisms requiring sustained ATP input are working tirelessly in the background just to keep these intricate molecular systems primed for immediate, high-fidelity response?
The energy cost of just being ready to perceive the world is immense. That is the question that really underlies all the physiology we've discussed today, the constant maintenance of a poised system.
A truly thought-provoking conclusion. The energy required to simply wait for a signal is as vital as the signal itself. Thank you for guiding us through the profound molecular basis of perception today.
It's always a pleasure to dive into the core principles of biochemistry.
And thank you for joining us for this deep dive. We'll catch you next time.